Hello, my name is Catherine Eckel, and today I will be talking about the article titled Species-Specific Bioluminescence Facilitates Speciation in the Deep Sea Breakdown. And this article was by Matthew P. Davis, Nancy I. Holcroft, Edward O. Wiley, John S. Sparks, and W. Leo Smith. So a little bit about the authors. The first author is Matthew P. Davis. His research focuses on evolution of fish in the deep sea. He was an associate professor of biology at St. Hosted, and he hosted a podcast called What the Fish bi-weekly from 2012 to 2013. He currently works at Cloud State University in Minnesota, and he was a research scientist at multiple museums as well. And then the second author we're going to be talking about is W. Leo Smith. He was an evolutionary biologist specializing in phylogenetics of fish. He also appeared on What the Fish, and he works at the University of Kansas and the Biodiversity Institute. So now a little background on our article. An important thing to know about this is bioluminescence, and bioluminescence is a product of a biochemical reaction where energy is converted into light. And some of the uses of bioluminescence is communication with other species or organisms within their own species, or to defend themselves from predators, or to attract other organisms in their species in order to reproduce. They are seen in vertebrates, mainly being cartilaginous and bony fishes, and those fishes are mostly deep sea organisms. Now, few studies actually look at the patterns and mechanisms of diversification in the deep sea because it's such a extreme environment that is hard for one to get to or do research in for that matter. So in this study, they are looking at lanternfish, which are the most species-rich marine teleos, which is a ray fin fish. And they will also be looking at bristlemouse, which are the most abundant vertebrates on Earth. And both of these fish are similar in sizes and body plan. So the research questions that they are asking is if bioluminescence mechanisms contribute to reproductive isolation and diversification. And the reason they're doing this is to provide some new insights into how bioluminescence might affect genetic isolation and diversification. And also they are doing this because there's such little research done on the deep sea in general. So first we're going to talk about the first figure, which I'll show in just a second. And the hypothesis that they were asking here is if there's a relationship between bioluminescence and evolution in deep sea lineages. And the way that they collected data was they constructed a temporal phylogeny using nuclear and mitochondrial data. And then the way they analyzed it was by putting it into a Geiger module in our platform, and then they calculated a 95% confidence interval of the expected number of species within a clade. So here you can see the first part of the figure. Right over here, this is the phylogeny that they created. So right here, this is pretty much the ancestral species. And over time, we have nodes branching out, so we're getting some more species over time. And in green and blue, green is our primarily ventral bioluminescence in the ventral photophores. And then with blue, this is pretty much if they have lateral and ventral bioluminescence. So as you can see with the ventral, that it actually showed up in 4 different places and evolved over time. And when they did show up, we did get more species after that. So maybe there is a link to bioluminescence and species richness, which we'll find out soon. And then here is the second part of the first figure, which shows that there is an increase in species richness over time. And then in our second figure here, the hypothesis that they are asking is if there is patterning of lateral and ventral photophores in Lampan Sitinae. And the way that they collected this data was simply by taking photos of the patterns with cameras and putting them into an analyzer. And the way that they analyzed this was putting them onto a graph with a TPS VIG version 2.16, and then they did a relative warp analysis. So this isn't as important to know. We just want to know that they put it into a computer program which analyzed the data for them. And on the table here, we can see that it was the two warp analyses were compared. And they did this by species within the subfamily Lambancidinae. And here on the left, we're looking at just lateral photophores. So as you can see, there is some grouping of where the lateral photophores are in this subfamily. And then over here, it shows the ventral photophores in the same family. And we can see a somewhat similar trend, but not so much in some of the species here. Some of it's kind of spread out and mixed around. So definitely more so over here in the lateral photophores. And then in figure 3, we're pretty much asking the same question, but with a different subfamily. So it's if the patterning of bioluminescent photophores in diaphis are different. And the way they collected the data was the same. They took photos of the patterns with their camera, and the way they analyzed it was the same by putting it into that program. And the tables look similar as well. So as we can see here on the left, we're looking at the lateral photophores of diaphis, and there is definite grouping of the patterning of the lateral photophores. It's much more clean than these over here. However, with the ventral photophores, it is much more messy, much more spread out. So we're seeing a little bit of a trend of the lateral photophores being more grouped together than the ventral. In figure 4, this one is much like figure 1, as they're asking if there's a relationship of forms and the species richness over time. And the way they collected data was also the same. They did a temporal phylogeny reconstruction, usually using nuclear and mitochondrial data. And the way they analyzed it was the same as well, by using a Geiger module in the R platform, and they calculated a 95% confidence interval of the expected number of species within a clade. So as you can see here, this is the first part of the graph, and we have time on the x-axis and the species on the y-axis. And here we have the ancestral species, and we see it slowly branching at first, but then more recently, there more species are showing up. So in this second part of the figure here, we can see a more simplistic way of looking at this, that the species richness is indeed increasing over time. Not as sharply as in figure 1, but it is definitely increasing. So the main findings that they had here was that the lanternfish photophore system had 2 functional roles, which was camouflage and species recognition. And the bioluminescence can act as a mechanism for genetic isolation. So if we're talking about species recognition, that makes quite a bit of sense if you look just at these, that because the patterning of the photophores was separated out compared to other species within the same family or genus, that it'd be easier for them to identify which one is within that same family, because they have the trend of being in this certain patterning. So, and then they concluded that it was a mechanism for genetic isolation, because if they're able to recognize species just by the patterning of their bioluminescence, then that means that certain species won't want to reproduce with others that have separate patterns from theirs. So it's going to isolate them from one another pretty much. And the way this relates to what we are learning is, because we have talked about genetic isolation before, mainly things like, ecological or functional isolation, like it doesn't fit or they're just separated by some environmental factor. But this is kind of a new one to us, where they are together and they are seemingly the same, but it's just a simple case of patterning that people aren't thinking of. So this article gives great insight into how much more we need to find out about genetic isolation and what other factors can contribute to reproduction. Thank you.